Hi, I'm glad that you made it back for this second part of this series, and today we're going to discuss a few more advanced applications and advanced topics of recombinases, and more specifically we're going to discuss recombination-mediated cassette exchange in more depth. And RMCE was first called double reciprocal crossover because that describes it actually very well in terms of mechanistics, and instead of having one crossover or recombination event, we have now two recombination events simultaneously, hence double reciprocal crossover, and we exchange one sequence for another. And that's pretty much it. So you could turn off now, but if you want to have a more fundamental understanding, and if you want to know how spacer mutants are used to design orthogonal recombination site pairs and how we use them for cassette exchange, then stay tuned and let's have a look at it together. In the previous video, we learned how to integrate, excise, and invert a sequence of interest using one pair of recombination sites. And this is super useful, for example, when we want to integrate a plasmid into the genome, because it is efficient and it's also very precise, so at one specific location. However, there is one problem. And when we look at our plasmid here, what is usually the case is that your sequence of interest might be this blue box here, but you have a lot more sequences surrounding it that you not necessarily want to integrate into the genome. And this is because usually you have things like antibiotic resistance genes or origin of replication that is just necessary for replicating or making more of a plasmid in bacteria. And not only is it a sequence that could affect your gene of interest or sequence of interest, but it could also lead to silencing of the whole construct once it's integrated, because it is a prokaryotic backbone. And to avoid that, we need to think about a strategy how to remove the bacterial backbone. And we learned about such a case, or we learned about a strategy how to remove something, and that is simply with a pair of recombination sites. So we can use a second pair and place them head to tail such that they would then excise the sequence in between. And so the idea is now that we can integrate the plasmid, yes, but we can also, in a second step, remove the bacterial backbone. So it will be excised. And at the end, you're left with only your sequence of interest. However, there is an important requirement for this, and maybe you've seen it already. What is not allowed to happen is that one of these red recombination sites is recombining with one of the black ones, because then what could happen is that, let's say, the sequence of interest, this blue box here, is removed and the bacterial backbone is integrated. So the requirement for using two pairs and performing integration and excision is that you have two pairs of orthogonal, non-cross-reacting pairs of recombination sites. And there are two ways how you can achieve that. Either you're saying, I'm taking two different recombinases, such as Cree and Flip, and use those in this manner. Or, and this is what we're going to focus on now, is we can use a different strategy. And we learned about, in the last video, we learned about mutations in the binding site of recombination sites and how we can then favor, for example, the integration over the excision. And now we learn about a second type of mutation, which is mutations in the spacer of a recombination site. Because recombination sites have a very cool feature, and that is that the exact sequence of a spacer doesn't really matter as long as it's the same sequence as the second recombination site that it recombines with. So what we can do is we can design spacer mutants. So we can take a pair and mutate the spacer here, and the same for the second site, and they can still recombine. but they cannot recombine with a different spacer sequence. So either with the wild type, or let's say we mutate also the second pair, and in this way we can generate pairs, many pairs of orthogonal non-cross-reactive recombination sites. So in this case we can cross out the arrow. There is no reaction between these two pairs of recombination sites. So we are very close at understanding how cassette exchange ultimately works, but we need to do one thing before, and that is discuss which pairs of recombination sites we should use. And we can use either Cre and Flip together, so two recombinases, or we use spacer mutants and just one recombinase. And of course, it would be better if we could use one recombinase, because it's just easier to express one rather than two recombinant proteins in a cell. And so what is the difference then exactly? And so let's think about using just one recombinase. What is the problem? The problem is that you cannot decide, or that you cannot regulate, which of the recombination steps happens first. So there are two options. Either you start with integration and excision of the backbone, like we discussed, or, and I added this now to our previous drawings, what could happen is that the excision happens first, and then the integration. And you can see that is perfectly fine. That can happen. In fact, there's a technique called mini-circles that uses the same idea, that we can remove the prokaryotic backbone of a plasmid and then integrate them or do something with them. And so this is perfectly fine. We just cannot really regulate which one happens first. But when is it not fine? And that has to do with efficiencies. So we discussed that integration is very unlikely, and that we can enhance integration over excision, but overall integration still stays unlikely for numerous reasons, such as that you need to get the plasmid into the cell, so transfection efficiencies. It needs to go into the nucleus. It needs to find the site where it recombines. So it will stay very unlikely, and the efficiency is low. So what people do usually is they want to enrich for the integration before they remove the backbone. So they use the backbone here for adding a selection gene. So what we can draw here is another gene, and I call it now S here, which is a selection gene with which we can enrich for the cells that have this integration first, and only then remove the backbone, which is so efficient that you don't need to select for it really. And this is obviously not possible. You cannot select if you have both ways going on, because in this case you will never have a selection gene in the genome. However, there is a way in which we can select for successful integrations, and at the same time we do not have to worry about the backbone, because it doesn't matter to us. And this will be cassette exchange. So let's talk about the final step. How do we get from the system that we had before to recombination-mediated cassette exchange? And there's really just one step, a tiny step, that we need to think about. And this is the most important moment, because if you understand this, then, only then, you can connect everything from before to what cassette exchange ultimately means and all its advantages. And the step is the following. Let's think about the attachment sites here in red. Their only purpose is to remove the sequence in between, to remove the backbone of the plasmid. That's all they do. And so what we can do now is think about first integrating the plasmid, then we're here. And what if, so let's look at this recombination site here in red. What if we would just take this recombination site from the plasmid sequence here and just already pre-install it also in the genome, because we do not need it first. It's actually a bit of a pain for us, because we don't want side reactions, right? We want to control what happens in any step. And so it's actually good that we take it and put it already here into the genome, and so it's not part of the plasmid anymore. And that is all we need to do. And so if we look now here, this is the cassette exchange strategy, so it's the redesigned way we could do this. And the only difference is I have a selection gene here, or a reporter gene, and we'll come to this in a second, but we moved the red recombination site into the genome already. And now everything works the same. The only difference is we have to start with the integration first. There's no excision, that's not possible, because these sites are orthogonal, they don't cross-react. We go to the integration, and in a second step, we now remove the backbone, and you already see what's happening here. Because we moved this recombination site already here, everything that is in between the two recombination sites is also removed. So we can, and this is the important part, it allows us to pre-install a reporter gene or a negative selection gene already in the genome that will also be removed. So we can install a reporter that reports us on the absence of the landing pad and on the successful exchange. And that's why it is an exchange, because we exchange the reporter for our sequence of interest. And so we can appreciate how this works here, but there is another thing that is going on, and that is, of course, now not only the black recombination sites can recombine, but also the red ones could recombine first. And this is actually not a problem, because if you look down here, the arrangement is a bit different. Now the red ones are taking over the integration, and the black ones take over the excision, and the end product is, however, the same. So this is not only not a disadvantage, it's actually an advantage, because we talked about integration being the rate limiting step, but now we have two possibilities of integration, and it doesn't matter which one happens. And this means that we would increase the probability of integration happening. And because we now forced an integration to happen first, so we eliminated the possibility of an excision, means that we don't need to worry about expressing Cre and Flip in a timely fashion subsequently after one another, but we can co-express them, or even better, we use the same recombinase for both steps, and it will always happen like this. And there is even another advantage, that we can now remove the selection gene here from the backbone. Why is that an advantage? So the selection gene that we had before here for enrichment, we don't need this because we have it now here, although this is a negative selection gene or a reporter gene, and this would be a positive selection gene. So here we select for the presence of the backbone, and here we select for the absence of the landing pad or the reporter gene. So why is it so nice to remove now this selection gene from the backbone? Because we also make the plasmid smaller, and a smaller plasmid usually has, for example, a higher transfection efficiency, and this again helps us with the overall integration efficiency at the end, because if there's more plasmid around, then it's also more likely that the integration will occur, which is the rate limiting step.